Hi everyone, in this video I'd like to be going over proteins. Mainly this will be a preliminary intro into the structure, so primary, secondary, quaternary, tertiary structures. Also where these structures are derived from, so peptide chains, long peptide chains, how they're built up from amino acids, what the basic structure of an amino acid is, like which functional groups are inside of it, how they're bond together. Also, we like to talk about the function, so what proteins do. Obviously, they're very important to everyday life and everything, but it's more than just, knowing how they're made. It's about knowing what they do, and obviously the origin. So like all these macromolecule videos, it's really important to know how we get them into our body. And so yeah, let's move on with the lesson. So next up here are proteins, and it's really important to know that proteins actually are really major function for a lot of things in our body. So actually, I like to just go over the function first because proteins basically do everything. Like they are one of the most important macromolecules in our body. And so an example would be that they function with our hormones. So the release of hormones, the carrying of hormones, DNA replication, DNA regulation, transport between cells, immune system, proper digestion. Just so many things in our body are governed by proteins, and so a lot of the current research is done around proteins and how they kind of interact with our body, with our DNA, etc., etc. And so the general structure of proteins can be dumbed down to the very simple basic building block of it, which is the amino acid. So this right here is called an amino acid. You can kind of see derivatives of where this name comes from because you kind of have like an amine group right here, and you have a carboxylic acid group. So you kind of just see how they kind of join the name together to make amino acid. Now, this structure, everything above this these are all the same for every single amino acid. Now, this R group kind of determines what this amino acid does. So in this case, an R group can be positively charged, negatively charged, hydrophobic, hydrophilic, polar, nonpolar, aromatic. You get the gist. All these have different functions. There's a lot to cover in those. I can have a separate video on how those things can be covered. Like I can write out each amino acid and kind of go over which one is which, like as in which property can kind of be encompassed by just looking at the R group, and how it can kind of function within the protein and how it kind of does its job when it's kind of inside that protein in the structure, and doing its thing. Now, it's important to know that proteins aren't just free floating amino acids. They're not just like floating globs of amino acids. Like they're structured, and so it's important to know that proteins are made up by 4 structures. In this case, it's primary, secondary, tertiary, and quaternary structure. And so I like to go into detail on what these structures are. So this primary structure here, we have a primary structure. And this structure is just a lot of amino acids just kind of bridged onto each other. So you can just amino acid attach to another amino acid. And then I'm just going to abbreviate to AA. So amino acid, blah, blah, blah. And then the bond that's formed between these two is a bond between the amine and the carboxylic acid. So you have an amine of 1. So this is a amino acid 1 plus the COOH, or the carboxylic acid of the other. And they kind of have a carboxylic acid addition reaction, and you come out with something that looks like an amide bond. So an amide bond would look something like this. So like this. And you have A1, A2. I kind of drew it flipped, but you get the point. This is the main bond between 2 amino acids, and we call this the peptide bond. That's the primary structure. So you can imagine the primary structure is just a long chain of peptide bonds between amino acids. Now, secondary structure, we have something called alpha helices and beta sheets. And so you can imagine once you have this long chain of amino acids, you want them to kind of form a certain type of 3D structure so they can be later formed into the globs that can actually be functional in the body. So in this case, alpha helices, well, it's kind of in the name, but they kind of make like a helix structure. I'm sorry, I'm really bad at drawing these, but they kind of look like, you can imagine like this, and then they kind of like intertwine with themselves. So this first one's kind of bad, but this one kind of makes sense. And these are kind of like built upon the interactions of the R group. So it can be like hydrophilic, not hydrophilic, but like polar, non-polar, hydrogen bonding, etc. All these kind of interactions pull themselves together to make these different types of 3D structures, because they're kind of attracted to each other and also repelled against each other in comparison to like other amino acids. And so these interactions of like, dislike, so like opposites attract, and obviously things that are not the same do not attract. Sorry. I'm going to cut that out real quick. So whoever's editing, just retract to maybe alpha helices. And now, so alpha helices are obviously made with like say polar interactions or non-polar interactions. So two things like, so two things that are the same, and two things that are not the same would obviously be more likely to be repelled from each other because they don't share the same kind of chemical properties. And same thing with beta sheets. So these things are kind of just like, imagine like instead of having an intertwining helices, you kind of have like peptide bonds just kind of sheeted onto each other, and it looks like kind of like a big flat sheet of peptides. And so these are this is like kind of the secondary structure, because you once had a linear form, which was primary. Now secondary, you're kind of having a more complex structure. And then now we get to tertiary, where you actually start to form the globular structure of a protein, which kind of looks like this. And these are driven by hydrophobic interactions, mainly because you want to have the infolding of proteins to make this 3D structure. And so that's where the majority of that infolding comes from, which is hydrophobic interactions, because the surrounding water kind of makes it so that this protein kind of folds into itself and makes kind of like a globular structure. Yeah. And then the quaternary structure is kind of just a higher order where a lot of proteins kind of work together. And so an example would be like something like hemoglobin. You kind of have like, say, like you have a protein here, you have a protein here, or something like that. And then you have different types of compounds or other proteins that help out. So you can see that it's a higher order because it's not just one protein anymore. It's a lot of different compounds working together to do one job. So now we've completed the structural and functional component. I think it's good to go over a practice problem. So let's move on to the practice problem and see if you guys can answer from what we've learned today. Okay, so these are the 4 questions that I've pointed out. And so take a moment to pause and look over these questions and then answer on your own time, and then unpause and kind of correct it with me. All right, so let's go over these questions. So what functional groups are on an amino acid? Like I said, we have an amine group. Looks like this. And then we have a carboxylic acid group. And that would be COOH. What is the purpose of an R group? What are the different types? Well, in this case, an R group is to differentiate each amino acid. Basically, it just gives it its function. So let's just say amino acid function. And what are different types? Well, you can just talk about how they're polar, non-polar, hydrophobic, hydrophilic. And you can also talk about how they're charged, negative, or positive. All right. Okay. Another one. What bonds link amino acids? Well, like I discussed, you have an H2N attack a COOH, and that's an amidation bond, or an amidation reaction, so you would have an amide bond. Something like this. This is an amide bond. And lastly, how many structures are needed to make a full protein? In this one, I just meant the number of stages. So you can say 3 to 4, depending on the order needed for work. As in, some proteins don't need to have in higher order, but some do, so 3 to 4 depending on that. All right, so it looks like if you guys got through those practice problems, I think you guys have a pretty good basis knowledge of proteins. Next up will be DNA and RNA, and we'll be going over their different structures and how they kind of play a role in our genetics. And as always, thank you guys for watching this video. Please check out our other videos at Chem Talk. We go over Gen Chem, organic chemistry, and biochem, of course. So please check out your other videos if you guys ever need help on any of those topics. And yeah, I'll see you guys later. Thank you. Bye-bye.